Assalamu alaikum. Today the topic of discussion is development of the musculoskeletal system. The lower limb bird develops on the 28th day, while this lower limb bird is converted into the flattened foot plate at the 40th day, and this lower limb bird will be converted into the sagittal equinus orientation on the 46th day. I told you about the development of skeletal system in my last lecture. In this lecture, we will discuss the development of the limbs. How the limb bird develop? Mainly they develop as an outpocketing from the ventrolateral body wall of the embryo in the beginning of the 5th week. In this 6th week, the terminal portion of the limb bird become flattened to form the hand plate and foot plate, and are separated from the proximal segment by circular constriction, the future wrist crease and ankle crease. In this diagram, you can see the embryo at 5th week. In this diagram, the embryo is at the 5th week. You can see here the upper limb bird and the lower limb bird. Then at the 6th week, you can see here there is a constriction of the hand bird, upper limb bird with the remaining part, and digital rays has developed. And now at the 8th week, the hind limb birds are less well developed than those of the forelimbs. Initially, they consist of a mesenchymal core. As you can see in this diagram, this is a mesenchymal core. The mesenchyme in the birds begin to condense, and by the end of 6th week, the first hyaline cartilage models foreshadowing the bones of extremities can be recognized. The endochondral ossification of limb bones starts by the end of the embryonic period. This is the diagram where you can see here, this is the upper limb bird. This is shown in the larger diagram. This pink is the loose mesenchyme, which is overlaid by the ectoderm. This is the structure of apical ectodermal ridge. And after that, this loose mesenchyme is condensed. Now this purplish is the condensed mesenchyme, and they will form the primordia of the forearm bones. After that, you can see here after the development of a cartilaginous model, the bones will ossify, and there is the development of upper limb bones, which are humerus, radius, ulna, and the carpus. And in this diagram, you can see the fully developed upper limb. Now comes the rotation of the upper and the lower limbs. During the 7th week, the upper and the lower limbs rotate in opposite direction. The upper limb rotate 90 degree laterally. This one is the upper limb. It will rotate 90 degree laterally so that the extensor muscles lie on the posterior surface of the upper limb, and the thumbs also come to lie laterally, while the lower limb rotate 90 degree medially so that the extensor muscles come to lie on the anterior surface of the lower limb, and the big toe lies on the medial side. Now comes the skeletal system anomaly. There is an anomaly which is known as the Achondroplasia. This is caused by the disturbance in the endochondral or intracartilaginous ossification in the epiphyseal or growth plate of the long bones. The result is the dwarfism. So both extremities are short, but head is of normal size. You can see here the head is of normal size, but extremities are short. Now comes the development of the muscular system. The entire muscular system of the body develops from the mesoderm except the muscles of the iris, which are derived from the ectoderm of the optic cups. The cardiac muscles of the heart develops from the splanchnic mesoderm surrounding the primitive heart tube, while the smooth muscle of the GIT develop from the splanchnic mesoderm surrounding the gut tube. Development of the axial musculature. The somites develop from the paraxial mesoderm. As you know that from the paraxial mesoderm, the condensed bodies or wide bodies develop, which are known as the somites. These somites give rise to the skeletal musculature of the axial skeleton, body wall, limbs, and the head. From the occipital region down, somites form and differentiate into the sclerotome and the dermatomatome. You can see here the somites are differentiated into the dermatomatome and the sclerotome. What is myogenesis? This is the process of development of the muscle fibers. Cells of the myotome or satellite cells in the body wall and limb regions split and become elongated, spindle-shaped myoblast. In this diagram, you can appreciate the satellite cells, which are the cells of the myotome in the body wall and the limbs. These become elongated and spindle-shaped and form the myoblast. Now these myoblasts will fuse to form the myotubes and ultimately mature muscle. By the end of the 3rd month of intrauterine life, cross striations, a typical feature of the skeletal muscle, will develop. Formation of the anterior and posterior groups of the muscles. At the end of the 5th week, the musculature in the body wall divides into a small dorsal portion, the epimere, and a large ventral portion, the hypomere. We should know that there are two group muscles, which are a dorsal portion, which is the epimere, and other is the ventral portion, which is the hypomere. The nerves supplying both these group of muscles are also different. The nerve innervating the segmental muscle is also divided into two rami, dorsal primary rami and the ventral primary rami. The dorsal primary rami will supply the epimeres. This one is the dorsal primary ramus. It is supplying the epimere, which are the muscles of the back or the dorsal region. You can see here the extensor muscles of the spine. While the ventral primary rami supply the hypomere, means the muscles of the ventral body wall or the flexor muscles of the spine. Formation of the extensors and the flexors. The muscles of epimere form the extensors of the vertebral column, while the muscles of the hypomere forms the ventral and lateral flexors of the vertebral column. As this is visible in this diagram, epimere, from here the extensors of the vertebral column will develop, and this is the hypomere. They will form the ventral and the lateral flexors of the vertebral column. The hypomere splits into 3 layers. In the thorax, these layers are represented by external and internal intercostals and the transversus thoracis muscles, while in the abdomen, these are represented by the external and internal obliques and transversus abdominis muscles. Development of the limb musculature. The first indication of the limb musculature is found in the 7th week of development as a condensation of the mesenchyme near the base of each limb bud. This mesenchyme is derived from the dermomyotome cells of the somites. The upper limb bud lie opposite the lower 5 cervical and upper 2 thoracic segments. You can see here the upper limb bud lie opposite the lower 5 cervical and upper 2 thoracic segments, while the lower limb bud lie opposite the 4 lumbar and upper 2 sacral segments. As soon as the buds are formed, the neighboring spinal nerves penetrate into the mesenchyme. At first, they enter with isolated dorsal and ventral branches. Soon, these branches unite to form large dorsal and ventral nerves. Thus, in a grouping upper limb, radial nerve, which supplies the extensors, is formed by the dorsal segmental branches, while the ulnar and median nerves which supply the flexors are formed by the ventral segmental branches. Now comes the dermatomal pattern. The spinal nerves not only play an important role in the differentiation and motor innervation of the limb musculature, but also provide the sensory innervation for the dermatome. Although the original dermatomal pattern changes with growth of the extremities, an orderly sequence can still be recognized in the adults. You can see here the dermatomal pattern which will be changed as the extremities develop. This was all. Thank you.